All righty, folks, we'll go ahead and get started. Thank you all for being here. It's my pleasure to introduce today's speaker, Dr. Dean Morales, visiting us from Los Alamos National Laboratory. Dean's a staff scientist 3 at Alamos. So I say that for a very important reason. So again, we're trying to prep our students for potential careers like national after. When you're a staff scientist, that means it's like the equivalent of being tenured. But it's a really good thing. Really good thing. It means they like you and we're keeping you. Very cool thing. Dean grew up out in the LA area and attended the University of California, San Diego, getting his bachelor's in molecular biology, and then moved on to UCSB and made a shift, something that's very familiar to me. He started moving into chemistry and went there to work with Norbert Wright at UCSB. And after completing his PhD, moved to Los Alamos as a postdoc, working with NPA Synth and some colleagues, Jim Werner and some other colleagues over in the B division as well. So taking advantage of one of the great things that National Labs do, which is collaborate well across lines. And his work really, his variety of efforts, including ranging from soil microbiomes to biofuels, to nanoparticle conjugates, to the molecule detection and tracking. So a lot of area covered. And again, I really resonate with that career path starting in molecular biology, moving his way through biophysics, biomaterials, and really excited to hear more about his work. Today he's going to be talking to us about his work with Plasmonic Nanoparticle platforms for your spatiotemporal controlled delivery of biomolecules. So with that, I'll hand it over to you, and thanks for joining us, Dean.
Okay. Very welcoming. Thank you very much. And just first off, thank you, Gabe, for inviting me out here, and thank you to all my hosts for speaking with me today. Like Gabe mentioned, I'll be talking about there's this little thing over here. Is that mic? Oh, no, I think on this one. I apologize. Is this
Part of our hospitality.
Yeah. Is this my? Okay, maybe it is.
National Lab computers very well.
Okay, yeah,
it
it
should
is.
be there. Yeah.
All right.
Don't leave the meeting.
So confused. Okay, perfect. So I'll be speaking about this nanoparticle construct that I've been working with my former colleagues at UC Santa Barbara, with my former advisor, Norbert Reich, and my very close colleagues, Xiao Huang, Gary Brown, and Alessia Polaro, where we were taking this idea of whether or not we can control biological activity pretty much with a flip of a switch. And if you had that ability to have such precise control over your cellular systems, this would have great implications in various fields of biomedicine and biotechnology, specifically tissue engineering, very with precise control, the different cell types that you're differentiating your stem cells into, drug delivery, mitigating all off-target effects by only releasing your drug where it has to be, and the list goes on. Even creating cellular factories that work in tandem to make specialized bioproducts with synthetic biology. But you can imagine that if we had this ability, we can start making huge advances in basic research. And so I'll give a very brief outline of what my talk will entail. I'll introduce our plasmonic particle platform on how we're using these to control cell activity, and then go into very specific examples of us using this platform, having precise control over RNA interference, and also how we could use this for potentially light-activated genome editing. And I'll very briefly close with the work that I'm doing at Los Alamos National Lab, which is really looking into the fundamentals of microbiology. And so I added this slide just for the non-biologists in the room. To be able to alter a cellular activity, we need to really fundamentally understand the flow of information that occurs inside the cell. So just looking at this very broad central dogma of going from DNA to RNA to protein, we can imagine this as we're going from a very large plan of everything that goes inside on in your cell to an individual blueprint for your molecular machines, and then the production of those molecular machines or your proteins. So you can imagine if you can affect one or many of these steps, you can significantly alter the activity of your cell. And so the platform that I mentioned we're using is based on these plasmonic particles, and we're using the unique phenomenon of surface plasmon resonance, whereby this collective oscillation of electrons that occur on the surface of these metal nanoparticles have attractive applications in both bioimaging, delivery, biosensing, and even localized heating. And so I'll be focusing on how we're using these plasmonic particles specifically in delivery and localized heating. But depending on what kind of laser excitation source we're using, we can have two different outcomes. For example, if we use continuous wave lasers, we can typically observe uniform heating of our solution that these nanoparticles are suspended in. But if we use very fast, very high energy laser pulses, what we can actually see, or what we can expect, is dissociation of those surface ligands that are coating our particles. And this is where the strength of that delivery system really comes into play. Now, one additional feature that sort of is very important for biological applications is the idea that these particles, when they're irradiated, can generate these very localized nanobubbles that allow us to permeabilize or porate membrane barriers. And so oftentimes than not, when you try to deliver a sort of biomolecule or drug into your cells, oftentimes they'll get entrapped or sort of stuck inside membrane-bound vesicles or endosomes. And so this platform not only allows us to release biomolecules from the surface of our particles, but also porate entrapping membrane vesicles, releasing those biomolecules into the site of action or the cytosol of the cell. For our particle systems, we are focused on strong absorbers in the near-infrared region. Ideally, this is because in that near-infrared region, or especially in the near IR1, we have minimal absorption by water, and very few molecules absorb in that region, so we're not having any off targets. And our particles are sort of localized to be absorbed or to absorb at that near infrared region. There's many classes of plasmonic nanoparticles that are available to us. For example, particularly gold nanoparticles, hollow gold nanoshells, as you can see on the left, hollow gold nanocages and nanorods. For the remainder of this talk, I will be mainly focusing on the platform that we were using, which is focused on these hollow gold nanoshells. And so to make these particles, we're typically using a traditional galvanic replacement reaction, whereby the electrons from the silver sacrificial nanoparticle are scavenged by the gold salt to deposit a layer or shell onto these templates, and further addition of gold salt will typically make them porous. And so if we look at this spectrophotometrically, we can see the transition of the plasmon resonance of the silver nanoparticle absorbing well into the UV that shifts into red shifts into towards the near infrared or the red region as the gold nanoparticle begins to form. And so depending on the initial size of our silver nanoparticle template, from smaller to larger initial silver nanoparticles, we can see a red shift of these this plasmon towards the red. And so for our nanoparticle platform, we're typically working with particles that are approximately 40 nanometers in diameter, with approximately 5 to 7 nanometer shell thicknesses that are absorbed strongly in the 700 to 800 region. And I'll go more into that in a short while. But after we synthesize these nanoparticles, One of the challenges is to make these particles biocompatible so we can actually use it in our biological systems. We're exploiting a very well-known synthesis or assembly technique, which is using thiolated nucleic acids, which not only are going to be our passivating layer on our nanoparticle structures, but will also be the cargo that we're using to deliver into the cells. And this can be done a couple of ways. One is depositing them slowly or assembling these the self-assembled monolayer of DNA or RNA on the surface slowly with the slow addition of salt, or just by dropping the pH rapidly and seeing a quick assembly of these nucleic acids on the surface. A general schematic of how this nanoparticle platform is expected to behave is that we internalize our nanoparticles into our cell system by the process of endocytosis, whereby they are again being entrapped inside these endosomes. And then after irradiating these samples with light, we'll see that dissociation of that thiol-gold bond, releasing the payload, and as well as the vapor bubble formation, which will permit endosomal escape. And so just going back a little bit to how we introduce our particles in by endocytosis, we really just rely on biology to be able to transport these inorganic materials into an organic system. And so taking advantage of well-known receptor biology, we could take very specific peptide sequences. For example, this sequence here, which is rich in arginines and prolines, that have a very high specificity for specific receptors on certain types of cells. In this case, this arginine-rich peptide, RPAR-PAR, is specific for a receptor known as neuropilin, which is involved in angiogenesis inside that's overexpressed in certain cancer cells. And to demonstrate that we have this very tight specificity of cellular internalization, if we compete these particles to internalize either into a prostate expressing or neuropilin-1 expressing prostate cell versus a non-expressing melanoma cell, we could see in the figure below that we're observing preferential uptake of these nanoparticles labeled with a fluorescent dye in the cells that are not melanoma cells that are marked in red. So preferentially, we're seeing that. And if we look at just co-localization of these nanoparticles with, say, an endosome tracking dye, such as calcium, we can see in red the nanoparticles are fluorescent, and green is that endosomal marker. And we can see high co-localization of the two fluorescent signals, indicating that we are uptaking our particles by endocytosis. And if we take away that neuropilin-1 receptor using melanoma cells, we don't see that coincidence. Yeah.
Can I ask a couple of quick questions?
Yes.
So is that peptide the RPAR form?
Yes.
Is that synthetic or natural?
That's a natural peptide that's found, at least in the process of angiogenesis. This is a synthetically made peptide from a commercial vendor, but I can go into the chemistry of how we attach that to our particles in just a very short minute.
And then the scale bar, see it in the right.
Oh, 50 microns on the left side.
So is that intracellular? Or is it inside one single cell? Or is it...
The particles?
Yeah.
Yeah, they'll be inside, internalizing into a single cell. I'm sorry, this is a very poor image, but you can see, at least from this image, there's like some barriers here. These are the individual cells. I really apologize. So if you look at these melanoma cells, the sort of shapes that are outlined in red are typically what we see for a single cell. And like I mentioned, we're trying to acquire or achieve a very precise spatial and temporal control of the release of these biomolecules. So we are using conventional 2-photon microscopes to be able to draw or physically what regions of interest on a field of view of cells that we want to irradiate with our near-infrared laser. And so to demonstrate that, if we take a look at just one field of view of cells on the bottom, the monolayer of cells will be quite indistinct, but just fast forward, our shape of our cells are going to look sort of like this. Currently, before excitation, they'll be normally observed inside endosomes, sort of these punctate dots inside the cell cytoplasm. And we can irradiate our very specific regions, for example, demarcated by this red triangle, and then use the focal microscope to excite the particles in both in all X, Y, and Z axes. And so this movie is just going through that process of us irradiating those samples. And what we observe after irradiation is that only the cells that are exposed to light that were in that sort of red triangle show a release of a dye that was on the surface of these nanoparticles. So if we overlay the two images, just the complementary colors of purple and green, if there's no change, it should remain white, similar to what we're observing in the cells that were not irradiated. And in the irradiated samples, we could see that these punctate diffuse into these green cells. And because we have such fine control of what we're irradiating, we can make very specialized shapes or patterns, not only in the 2 dimensions, but in 3 dimensions. So this is just a image showing us drawing letters or trying to draw letters with our nanoparticle constructs, or even drawing cute shapes like heart. And like I mentioned, if we even use a 3-dimensional structure like a cell spheroid, we can begin to see that these nanoparticles with a dye, when irradiated across a 3D cell spheroid, we can begin to see a release of that dye inside these cells. And so now that we understand how this platform is going to operate, I'll go into very specific examples of how this technology can be quite useful in biological studies. And so the first example is looking at RNA interference, or RNAi. And this is through the delivery of a RNA duplex, also siRNA, or small interfering RNA. And when you introduce these small duplexes into cells, they'll complex with a protein to be able to identify its complementary mRNA target and be able to cleave that mRNA, preventing the synthesis of those molecular machines. So you're preventing the activity of cell using this technique. And so again, going into the sort of assembly of these particles, we're doing a self-assembly monolayer of the single-stranded RNA on the surface, and we could hybridize the complement to complete the siRNA duplex by adding the antisense RNA to the nanoparticle, and then labeling a cell-penetrating peptide onto these, to the termini of these scaffolds. And what we observe, at least with the dihydrodynamic radii of these particles, is that we're increasing in size as we're assembling these nanoparticles, but we also see a characteristic redshift of the plasmon as we complete the assembly of the particle construct. And so to imagine how we're going to be using this in cells, we're focusing, at least on our first example, the delivery of a potential anti-cancer therapeutic. This is polo-like kinase that when you knock the activity of this RNA down, it interrupts mitosis or cell division and causes cell death. So we're delivering our protein or our RNA, siRNA, radiating with light, releasing the siRNA, and causing cell death as a potential therapeutic. And this release of RNA on the surface, we observe to be quite power dependent. So this is 10 seconds of irradiation with various power densities for the near-infrared light. And we can see a majority of our RNA that's on our surface is irradiating at some point, or is being released at some point. So using those parameters and introducing these into cancer cells, the main takeaway from this very busy figure is that when we combine the nanoparticle construct with its targeting peptide and irradiating with light, we can see a significant drop in cell viability over the course of the treatment. And mind you, this is only a single radiation event that occurs at time 0, and we're seeing how little the cells begin to divide after 72 hours. This is quite comparable to commercial reagents that are used to transfect these siRNA duplexes into cells. This is a sort of detergent-based system known as lipofectamine, and we can see that our nanoparticle construct has quite comparable results. Most notably in column D, if we don't irradiate our sample, we see that there's no shift in that viability, meaning that these particles are quite contained and don't cause any off-target effects when not treated with the laser excitation. And even if we remove the targeting peptide, whereby the particles are unable to access the cell, by removing that, we prevent any cell death just by preventing any access of these particles into cells. And just to show that it's not just laser excitation and the particles that are causing cell death, just as a physical mechanism, if we look at the expression of that PLK1 gene that I mentioned earlier and do PCR where we amplify that gene and look at just how many copies of that gene is present, if we compare it to the sample that was not irradiated or even just the cell-only control, we see that PLK1 gene really being downregulated and causing that cell death of these cell systems. And like I mentioned, these particles are very specific based on that arginine-rich peptide. And if we take a prostate cancer cell, or not even prostate cancer, but a prostate immortalized cell that does not overexpress that neuropilin-1 receptor, we see there's no access to the protein or particles into these more wild-type cells, and that after irradiation and through treatment, we don't see a change in viability. So this is providing us so much control on many different levels. We can imagine that by expanding our horizons and what this technology could be used for, like I mentioned earlier, we could potentially see some value in being able to use this to engineer different tissue types with very precise control. So if we take another construct of these nanoparticles and attach another cell-penetrating peptide known as TAT, this is a peptide derived from HIV. It can give us global entry into different cell types. And so by introducing this siRNA duplex again into human embryonic stem cells, we could imagine if we downregulate certain transcription factors or certain factors that allow these cells to differentiate into different cell types, we can control the activity of the cell and differentiate our cells into what we want and where we want. So just very briefly, if we take one siRNA for a specific Yamanaka factor, OCT4, and downregulate that, what we see or what we have observed is that these human embryonic stem cells will differentiate into pretty much all three germ layers that are required to further differentiate into different cell types. So this is just the beginning of what this technology can afford us. So moving on to more like this pop science technologies that are now available to us. I'm pretty sure many of you are familiar or have heard of genome editing, especially with the advent of humanized Cas9 and being able to deliver these enzymes into human cells. We had the original idea that we can use this platform to elicit genome editing in only the cells and when we want these cells to be edited. Now, to introduce our protein synthesizers, there's several different mechanisms, but proteins are like I mentioned, your molecular machines, and they'll have direct activity inside a cell once you deliver them into cells. Unfortunately, that means they're quite sensitive to different environmental effects. So the nanoparticle platform gives us this ability to release them by retaining their activities. And once we deliver them to the cells, we can expect specific activities to occur, allowing us to apply this to different fields of biology. And so to assemble different enzymes and or peptides onto these nanoparticle constructs, we have a very similar construct that I've been showing you already, but we're including this very specific moiety at the terminus of our scaffold, which is based on a nitrilotriacetic acid. Now, I'm not sure if everyone's familiar with traditional biochemistry and being able to purify proteins, but if you took an early biochemistry lab, you'll have been familiar with different engineering strategies or protein engineering strategies to include these sort of specialized peptide tags at the ends of your peptide to be able to complex them with this nitrilotriacetic acid moiety in the presence of a divalent cation. So this is a very convenient way to virtually assemble any protein onto our nanoparticle construct. And so to demonstrate that we could be delivering these proteins into cells with spatial control, we used a green fluorescent protein, again, that's attached to that NTA moiety, and also expressed this with again, this arginine-rich peptide to internalize them into cells. And looking at just before and after release of these green fluorescent proteins in these cells demarcated by these red diamonds, we can see before and after release, we're seeing that diffusion of GFP into the cells, into the individual cells, only when we're irradiating them with light. If we use a higher magnification onto the individual cells here, we could see that the GFP nanoparticles are trapped inside endosomes, and then after release, we see that diffusion of that fluorescence from that puncta to sort of normalize across the entire cells. And so, like again, to show that we wanted to deliver an enzyme with certain activity, we're looking at genome editing machinery to either knock out or delete genes, or even introduce new gene functions into cells. And so the model that I'm using or have used on this system is this enzyme known as Cre-recombinase that when it recognizes certain sites that flank our genes of interest, known as LOXP sites. These are just sequences that are flanking our DNA that we want to rearrange. It'll recognize these sites and then cause certain rearrangements of the genome, depending on how those LOXP sites are arranged. And again, going back to this very familiar cartoon now, we're using this Cre-recombinase, so it has that, again, TAT peptide to give us global access into different cell types. And we're, again, complexing these to our NTA moiety and delivering it through this process of endocytosis and release from the nanoparticle. The proteins themselves, or the enzymes themselves, do retain their activity when they're on the surface of these nanoparticles. If we take our pre-labeled particles and then just dissolve away the gold nanoparticle platform from this, we can see on the very end, especially here, we can see the activity of the Cre-recombinase in action, where it takes this linear sequence of DNA and then rearranges it to make a circularized plasmid that's formed at the above here and below, and also excising this very small DNA fragment, which is observed here. So when presented on the surface, we can retain the activity of these proteins. This is especially a concern because these enzymes can be quite sensitive. And delivering these into cells using TAT recombinant or TAT peptide, we can have concentration-dependent uptake of these nanoparticles inside cells. So here we're just using dark field microscopy to show these nanoparticles in these orange dots. And then to be able to demonstrate that we can retain the activity inside cells once the pre-recombinase is delivered. We're using a fluorescent reporter for a red fluorescent protein that has a sequence within its DNA that interrupts the transcription of this gene, so preventing that blueprint from being formed. And then pre-recombinase will be introduced into the cells to repair that activity and then allow us to express that red fluorescence again. And so just looking at the cells alone, we see that this reporter is not active. Very few cells show any red fluorescence. And again, if we introduce the gene using commercial alternatives, such as that detergent reagent, we can see it upwards of 24% of these cells can activate this recombinase reporter. And now, if we use our nanoparticle system that has this recombinase onto our nanoparticle system and irradiate them with light, we can see a shift in the population to cells that are expressing the red fluorescent reporter. And if we don't irradiate these samples, we see minimal or to no background red fluorescence being expressed. And so just a quick note, if these recombination efficiencies might look a little small, but if we take a look at just the commercial alternatives, we're only getting upwards to 25%. So if we normalize our release and activity of these nanoparticles, this is quite an efficient system for delivering of these genome editing enzymes. And again, we're trying to demonstrate both spatial and temporal control. So if we irradiate specific sites on a field of view of cell monolayers, only the cells that were sort of irradiated in that initial time period that's demarcated in these grids. If we just take a look at zoom in picture here, only the squares that we irradiated, and over time do we see that red fluorescent reporter being expressed. So this is a quite valuable system that we can imagine has great opportunities to be used in different genome editing platform or experiments or in tissue engineering. And so, like I mentioned, the last part of my talk will briefly look into my career at Lannel as a postdoc, where I took a short departure from materials to look at fundamental biology questions, specifically how we can understand fundamental interactions between bacteria and fungi that were derived from soil, and how we could use those relationships for very important national security endeavors, especially mitigating climate change or for the sustainable production of biofuels. And so the idea is that we're looking at fungi, a really underappreciated microorganism, especially since it's such a large kingdom. I'm sure that many of you have new familiarity with fungi with watching The Last of Us. That seems to be where everyone hears about it the first time. But these organisms are very abundant in soil, and we just don't appreciate how diverse they can be and how they interact with bacteria especially. I guess the main takeaway that at least our group has found was that if we take individual fungi derived from soil and sequence the entire genomes, not only the genomes of the fungus itself, but all the coexisting genomes that are related to it, so bacteria and whatnot. And these are, again, individual bacteria that are sort of isolated, separated from the source. And if we do very broad scale sequencing, we have observed that these fungi are coexisting or have very strong relationships with bacteria, archaea, viruses, and even some plastids. And this figure on below, this is just a very complex taxonomic representation or taxonomy, or your different levels of organisms, of how we classify organisms. We're looking at the different, very broad array of phyla from different fungi that are found from widespread of samples across the United States and from our own lab. And we can actually see that there's so many different bacteria that are associating with these fungi, something that's been completely underlooked or underappreciated to this time. And so another interesting facet of this is that not only are these bacteria and different microorganisms associating with fungi, but we can even find that these bacteria are actually living within fungi. So if we go back to a very fundamental idea in biology, this idea of endosymbiotic theory, whereby in our own cells, we've taken up our progenitor bacterium and made that into our mitochondria. This is a very similar interaction where these bacteria can coexist inside these larger, more complex organisms like eukaryotes or fungi like ourselves. And so the green here is showing the individual bacteria that are existing within these red mycelia of fungi. And if we just appreciate how expansive these interactions can be, we can see that the bacteria are almost ubiquitous across mycelial tissue. And so where do I see these efforts sort of marrying with my previous research experience? Well, as I mentioned, mycology, or the study of fungi, has been largely overlooked and underappreciated, which means that there's a lot of space for us to develop new molecular tools to be able to tease out these very intimate microbial interactions, especially from the ones derived from soil. So for example, in the idea of carbon sequestration, plants will pull carbon out of the atmosphere, and that decaying matter of different plant tissue will be degraded by different fungi and take that carbon and form this small organic acid known as oxalate. And what has been observed up to date is that the oxalate produced by fungi can be actually used by certain bacteria inside soil as well. So there's this idea now that is it possible that fungi are producing this secondary metabolite to practically recruit specific bacteria to be able to associate with their cells. And so one project that I'm working on right now is taking fluorescent quantum dots and labeling them with calcium, or labeling calcium oxalate crystals with this, and being able to see if we take quantum dot crystals and then present these to oxalate trophic or oxalate eating bacteria, will they eat the oxalate and then use the fungus as a means for interaction? So that's one avenue of where my research is being directed. And so I gave you a very global summary into my previous research endeavors, where I use plasmonic nanoparticles to acquire spatial and temporal control of activity of different biomolecules inside a cell or a site of action. And now I'm looking at how I could better probe microbial interactions to really understand how we can use microbes to better steer ecosystems, especially in the face of climate change. So with that, I'd like to thank my collaborators, especially the colleagues at UC Santa Barbara and Los Alamos. And I'll just leave you with one last slide. Again, I'm a scientist at the Center of Integrated Nanotechnologies. We're a DOE funded user facility. So what that means is that all our instruments that are funded by the DOE and expertise that's provided from our scientists are free to access to all our collaborators. So twice a year, we have a rolling application to submit a proposal to work with myself or many of my colleagues and give you access to this very valuable resource. So thank you very much for your time.
Time for questions, just for the record, without that slide, his Lionel badge would have been revoked immediately.
This was all a recruiting trip.
Yeah.
Yes.
Ended on citing nanoparticles with compared to laser. When you're actually putting it in the body and applying it to that application, how are you gonna do it? Gotta get it in.
Yeah. So I mentioned that we're using near infrared light. That's within the therapeutic, in vivo therapeutic window. It could get, I mean, at most like 10 centimeters into skin. I mean, this has always been this idea that when we originally went down this path, we were like, okay, can we make a therapeutic for this? Can we market this as potential drugs that we can use? And like you mentioned, this is the treatment itself is relatively benign. So the laser power and the exposure time that we actually need to release these payload into cells is, I'm trying to get back to that slide. It's quite benign to the cell system entirely. So even if we irradiate the samples that do not have the siRNA or C that do not have, one of these. It's always trouble to see the map, especially behind me. But the point is that if you take the siRNA, scramble that siRNA so that it doesn't recognize the peptide or the mRNA, it doesn't cause that cell death. So relative to even just a cell-only control, no treatment at all, we're having similar viability. So it's relatively benign to the cell system. But yes, the concern that if you wanted to use this in an in vivo system, there will always be more biological barriers. It's not as simplified as the cell monolayer system. But again, as I showed you before, even with 3D spheroids, we can irradiate these cell or this particle system in a 3D environment. So it does have the potential. It does need more research to be able to transition this. And oftentimes when you see a system like this, it's often used for photothermal photothermal ablation, where you're just blowing the hell out of your cancer cells.
Can I just say something, Brett?
Yeah.
It's pretty creepy to think that our eyes, if we could see a little bit further red than the neurobrain, we could look 10 centimeters, that's 4 inches, into your or my, anyone's tissue. It's creepy, until you feel creepy, or it's creepy. It's kind of like X-ray vision. And that's kind of a miracle in itself, that you're infrared. And it's hard to show. You can't show it with reds and greens. You could use false colors. But with our eyes, human eyes at least. Oh, sorry for that.
Yes.
Why do you have to use football open mic?
We had this commercially available to us. Otherwise, we would have had to build our own system. It had the software and the laser system, my TIE Sapphire laser, already ready to go. So we optimized it using that system.
Potentially, you can use a regular component.
Yes. As long as, so again, we're using ultra fast near infrared pulse laser excitation. So it does require the pulse laser excitation.
Yeah.
Yes.
Yeah, because what's your limitation there? What do you need for your pulses or femtosecond pulses?
Yeah, we need femtosecond pulses. Yeah.
That's a big thing.
Oh, yes.
Have you done anything that at least medical trials there, study, mouse study?
I personally have not. No. We've always entertained the idea. And at the time when we were doing this, it was hard enough to even get mouse models to do this work. So this is something that we just didn't have the funding to pursue any further.
I realized I know people's names and you don't.
Yes. So these are the theoretical excitations of our nanoparticles or gold nanoshells. So depending on the diameter of our nanoparticle, as well as the shell thickness, this is a shell thickness of approximately 7 nanometers, really governs where these particles will absorb light the best. And so, like I mentioned, we were trying to get these into the near infrared or 800 nanometer region. So that's why the size that we chose were ideal for this. Again, these are approximately 40 nanometers, but you can see a very broad absorption band centered around the near infrared.
Yeah, I had a question on it too, but you're able to keep your shelf thickness the same, right? And I think it's important for your plasmonic that you're getting out of that, right?
Yeah.
So you're 7 nanometers, but you're changing that, you're tuning in your wavelength just via the particle size.
Yeah. So we start with a very defined silver template. That's the factor we could control the best. And then we slowly add gold salt to cause that galvanic replacement. And we'll stop the reaction once these particles hit 800 nanometers. So it's a little messy. It's not perfect every single. Yes, gold, silver.
Shells, right?
Yeah, gold, silver shells. Yeah, and with the gold salt or gold chloride, oftentimes our silver ions will just crash out of solution. So we could clean up the system that way.
Other question?
The, there's like ecology. This is that, that's kind of like that.
Yeah. Yeah, I had a unique change of heart like halfway through graduate school. I was trying to figure out what I wanted to explore in my career. And one thing was for certain is that I wanted to pursue a career at the national lab. And at the time, the national lab wasn't looking at research like I was doing in with the gold nanoparticle systems. But what they did need was my expertise in single cell analysis and be able to tease out different biological events occurring at the single cell level. So there's a lot of work that I haven't shown, but a lot of it revolves around single molecule analysis of different biomolecules.
You know, the other thing about that is that, you know, his track and where he went to the school in UCSB, we mimicked a lot of what they do over there in terms of developing interdisciplinary type of approaches. So he was able to do that, you know, make those shifts. You're all, if you follow that path, you can make those type of shifts and being able to change like that's really important. Yeah.
Do the nanoparticles ever like change size while you're delivering them? Or
They'll change size when we assemble. If you recall that one figure, let's pull that up. They'll change size once we fully assembled the particle system with the DNA and even the cell-penetrating peptide. But these are quite shelf stable for long periods of time.
do you have a relationship in that size, wavelengths, that you know what you're shooting your laser at once you have that nanoparticle at that set size?
Can you rephrase it?
So yeah, so you have you're growing your nanoparticles, changing size.
Yeah.
But you're seeing a red shift as they get bigger. So like, do you have a relationship between those two things, or do you just have?
We take into account. So a lot of our particle systems actually start further blue shifted, and we expect it just from what we've empirically observed is that the red shift usually around where we want them to be. Does that answer your question?
Yeah, so it's empirical rather. You just have like a.
I mean, the figure I showed you with those very nice, yeah, plasmon resonances. This is all theoretical.
If you have that.
We could predict that. It's one thing to do it experimentally.
Oh, Rob, did you have a question?
Yeah. See, we have like 5 minutes.
Yeah, we got yeah.
You were careful not to show any experimental, like optical layout diagrams of how you do all this work, to keep your secrets or.
Oh, I mean, I couldn't draw you a diagram. This is the best diagram I can make of a focal microscope.
Yeah. Okay. There's your near IR pulse, which the plasmonic gold, hollow gold nanoshells orbing. And then
We're using a galvo mirror to sort of direct the light as it rasters across the
as
cells.
it rasters,
Mhm.
but then you have a separate optical layout system
That-
for your dyes, your green fluorescence
Right.
protein and whatever the red pigment name was.
Yeah.
And, so is that all using the same optical system?
It's using the same optical system. So it will, there's a shutters that will open up the near or visible lasers to probe our system, like the green fluorescent protein.
So you would have to superimpose
Yeah.
on your diagram where and it was 2 photon excited
Yeah.
fluorescence too, so that you can again penetrate deeply. I think that was Professor Jackman's question,
Mhm.
was why 2 photon excited fluorescence.
Yeah.
And I didn't hear you say it in the answer that, yeah, that's also, it's not quite in the window, but it's also penetrates very deeply.
Yeah. And then even in this figure, this is actually observed, this movie was looking at the particle continuum emission. So that's what we're seeing just after irradiating with the near infrared light. We're seeing that scattering across the nanoparticles.
Scattering from the plasma.
Yeah. So.
Even though you may not be in resonance
Mhm.
with it. Could you just go to, I think it was slide 41, where you revisited the whole workflow scheme? Or someone else has an urgent question. Yeah. But there's the 800 nanometer line up at the top
Mhm.
for activating, for releasing from the gold nanoshells.
Yeah.
And then, okay, so then the fluorescence diagnostic, and these are your bioconjugations.
Yeah.
So maybe it was
We're using green fluorescent protein, at least for
GFP,
one of our
okay.
markers.
The
Yeah.
GFP, the green fluorescent protein, is already attached to the
Mhm.
tool? No shit, okay. I'm sorry, I missed.
Oh, sorry. I was more trying to explain the modularity of the system.
Sure.
But yes, for at least our initial test, we use the green fluorescent protein to show that the fusion.
I had a question on your, so that stem cell differentiation, is pretty awesome.
Mhm.
What are your knobs on that? I mean, does like the particle or nanoparticle properties play in that, or is it just your siRNA tag in terms of driving, where you're going to actually differentiate to? How do you control
Yeah.
that?
So the problem with at least the OCT4 example we
Yeah.
used, that's a global Yamana. So that will just differentiate your human embryonic stem cells to, like I said, all 3 germ layers.
Okay, so,
So
okay.
you need further additions on top of that to differentiate the specifics. So it's never just a one-off
And you just
process.
engineer those in.
But we can imagine if we had different particle systems at different resonances, very spectrally
Right.
separated. We could have the same particle systems just having 2 different siRNA for different factors and release them. And again, I said that this particle system is also power dependent on how much it actually releases off the surface. So we can control just how much would be released to maybe make that effect less potent or more potent, depending on how much we radiate.
So can you, or has anybody been able to demonstrate this idea of actually generating like a 3-dimensional tissue by spatial localization and differentiation?
Not yet. So we stopped this back in 2017 or around 2017. We did it with GFP knockdown in these cell spheroids. We couldn't get to the point where we could actually start knocking down the different Yamana or the different transcription factors to do that 3-dimensional tissue regeneration.
Okay,
Tissue modifications.
that stuff's cool. I was jotting down those papers. That was really slick. You got it.
Let me make a comment. The last
Yeah.
part of very important, the relation between fungus and bacteria.
Yeah.
And let me give you a very important example. You go to the hospital and the doctor says the surgery was a success, but there was some complications. Means that you got an infection in the hospital and you probably have COVID.
Yeah.
And infections in the hospital happen by 2 things, the candida and then the bacteria. And when you take them, example, they always together. They live together. And but the doctor says they give antibiotics, kill the bacteria, and for some reason the fungus then, of course, they have more food to eat, but then the candida is 10% of the infection, 50% of the deaths in the hospital because of. So it's very important to understand this, and I've seen many pictures of bacteria and candida. I don't really understand the relation, why they live together, and probably you gave a very, but it's a very important problem.
Yeah, NIH been there.
That's.
Yeah. I mean, there's a huge effort now for fungal pathogens. Just this year, they came out with a top 10 list of just different.
Of course, the
pathogens.
antibiotic not goes with fungus.
Mhm.
Pretty morbid that you're doing.
Yeah, well, So, to speak on that idea, you might have, you missed this earlier, but I was explaining that this particular fungus that we're looking at, the bacteria in this is very dependent on the fungus. It's called Mycoavidus cysteine exogens. So it can't produce its own cysteine. So it takes it from the fungus. But what's very interesting about this fungus in general is that these, when this fungus is present on like rice, it actually becomes pathogenic if it only has the bacteria associated. You take the bacteria away, other fungi, rhizopus here, if you take that away, it removes the pathogenicity.
Which one is that?
This is Mortarella with a Mycoavidus, but this has been observed in different fungi that are related to this.
Very good.
You know, I was curious about what's the role of the QDs in that project? Are those just to visualize?
To visualize the transport. So
Okay,
okay.
okay.
There was
So
a
they're not like
study
anything
done out of Amsterdam where they labeled quantum dots with certain secondary metabolites, and the fungi will actually take these up and then transport them across long distances. So this actually goes back to this global idea that hasn't been proven yet, this idea of World Wide Web or Wood Wide Web, where all the trees are sort of connected by a mycelial network.
Yeah.
And so they've shown that you use different nanoparticle labels to show that transport of different molecules across distances. And so that's something that,
Some things that, yeah, you're not right. You know, it's funny, just side note before we end up, you put up your first slide, there's a picture of Gary Braun on there.
oh, you know,
Gary was a RU student when I first started as on our
Yeah.
team, when I was a postdoc at Atlanta, like 20 plus years
I think he
ago,
worked with
which is-
Andrew.
with Andrew, and I was like the new postdoc working with them. Well, that's just, wow, the world's connected.
Yeah,
folks.
it's
It's crazy.
my
It's
connection.
probably like the senior scientist getting there. Anyway, any other questions? No, let's thank Ian again. Thank you. Yeah, if you talk to Gary, someone said hi. Actually.
I will.
That was great. Awesome.
Yeah, my